So good afternoon, everyone. I'm Twinkle Soni, and today we are going to learn AMR analysis. So basically, in this, I'm just giving you a brief in this presentation. My main name will be on hands-on. So this is how we have learned till now, like how the data is up to the NGS, like any sample, human or animal sample or whatever sample you wanna do for analysis, you can take that sample. Metagenomic samples are generally preferred for AMR. Basically, we prefer metagenomic samples. If not, some projects are there which work on specific bacteria, like for Salmonella. A work in MSU is currently doing, like a group is doing to estimate AMRs in Salmonella specifically. So we can do such kind of things. Or else metagenomic samples, mean normal samples you can take. For AMR analysis, we generally prefer gut microbiome samples, like gut samples. So stool is basically preferred, or else blood. So DNA extraction and all the steps of library preparation which we have learned till now. It is just an introduction of that. So how exactly amplicon sequencing works? The sequencing output is there, and I guess quality filtering has been taught to you in AAU. So first you see you have learned till now. So after that, there are two types of tools which are available for AMR analysis. First is web-based tools, and second are Linux-based or standalone tools. So what exactly happens is web-based tools are generally preferred by normal people, but what exactly happens? The softwares which are on the left side, like one code is Cosmos ID, they both are paid software. So this one, like we have left. I will show you. I have learned for both of them, but we are not able to access them. Like for access, we need to pay. So it will be like not at a cost effective for us. Next is Rasfinder. It's good tool, like it's free of cost. But the problem is it will give the resistance against a particular bacteria. Like you can search a metagenome sample against a particular bacteria. I will give demonstration of all the things. Then you will come to know that what exactly I am talking about. Next is standalone tools. Basically, these tools are preferred. The only limitation is you need to learn the basics of Linux. And in some basic commands for blast and to analyze. The major work will be done in Excel. Apart from this, card, RASFAMS, MegaRAS, AMR Finder, all these are all available. What you need to do is you just need to download the databases online. You can download it online and make your blast database. There is a command make blast db. By this you can make database of any parser file which you have. So after that you can run your blast against that file. Blast exactly is just alignment tool. So it will align your file against the particular database. So it will do such kind of thing. Here is just a comparative for all of the databases. Card, Rasfinder, AMR Finder, Cosmos ID, and one codex. We will learn this via like training, not just by saying it. So I would like all of you to just log in. I'll let you know how to log in. So if you find any problem, do let me know. So, please. You need to type putty. What exactly I'm doing over here is this that we taught you all yesterday, that we have a high connection. everyone able to reach up to it? Right? Okay. In order to see whether you are in your working directory or not, like the directory of your design, you can always type pwd. So it will show you parent working directory, and it should be same as the address which is written over here. If not, then you are at wrong place. Cool. Is he able to do? Huh. Please feel free to ask. If you are not able to do until we did, then please ask. Cool.
आप इंडिविजुअली भी जा सकते हो और यू कैन डायरेक्टली कॉपी दिस पाथ फ्रॉम हियर, कॉपी पाथ टू क्लिपबोर्ड एंड यू कैन डायरेक्टली पेस्ट इट ऑन द टर्मिनल. तो डायरेक्टली वो वहां से लिंक हो जाएगा चेंज डायरेक्टरी. तो आपको पूरा पाथ टाइप नहीं करना पड़ेगा कि हाँ हमें अभी यहाँ पे है, फिर यहाँ जाना है, फिर यहाँ जाना है. तो दिस इज लाइक शॉर्टकट.
Cool. Now, as I told you, we have files in custom format when we download it. Over here, the nucleotide underscore library.fasta and protein underscore library.fasta. These two files are there on CAR database itself. You can download it directly. The main issue will be this file when you download these two files, nucleotide library.fasta and protein library.fasta. The main problem will be you need to create a database first. See, you can directly work on FASTA file, there is no issue. But what will happen if you directly work on FASTA file, you cannot give threads. So what exactly thread is? You are asking your computer, like server, we have only one CPU in our PC, normal PC, but on server we have like 250 or 230, something like that. So like CPUs are there. So you can ask 50 CPUs to do a task simultaneously. So it will be quick in process. If you are not doing it, like if you ask for, like if you do not create any database of these files, it will only one CPU can be allotted to a file. So it will take a lot of time. Moreover, if you are working on iron torrent data, the data will be quite high in the size. So it will take more time. So for that, we need to create a database so that we can give the threads to it. Moreover, database can be accessed through any path. Like if I am in data, I can access this database from Bhadesh as well. So there is no need to copy paste all the files in time. So database has already been created. So these files, such formats are there. Called the nucleotide underscore library.fasta is a database. So for now, I will tell you how to create a protein, like any database. So for that, it's a command make blast db. -in for input, and input file will be a protein file. Like for this, you can press P and tab. It will automatically take your file. Next thing that is mandatory is what type of database it is. So it is a protein database, so DB type. DB type. And write PROT, protein. Nucleic acid के लिए, for nucleic acid you need to write NuCl. It's a protein database, so PROT. Done? Is everyone able to write these commands? All right, now make class cd is the name of command. Hyphen input is the name of your file for which you need to create database. DB type is the type of database that you want to create. It is a protein database, so I write BROT. If it is a nucleotide, write NUCL, the first whole letter, and press enter. Done.
ठीक कहीं किसी को प्रॉब्लम हो तो प्लीज
Please feel free to ask. It's a hands-on training. Cool.
अभी आपने जैसा
Now,
वहां पे लिखा है वैसा आप मेक ब्लास्ट डी you are creating a database. You पता है you database create कर रहे हो आप लोग
just now do it.
कुछ तो पूछो database क्यों बना रहे हो आप लोग
डेटाबेस किस चीज का बना रहे हो?
डेटाबेस आप एक फाइल का बना रहे हो सी हम ब्लास्ट किसके अगेंस्ट करते हैं दो फाइल होती है हमारे पास एक क्वेरी फाइल होती है और एक द फाइल जिसके अगेंस्ट आपको ब्लास्ट करना है तो हम सिंपल उसको कर सकते हैं कोई इशू नहीं है हम अपनी जो जिस फाइल के साथ करने वाले हैं उसको हम सब्जेक्ट में रख के कर सकते हैं बट डेटाबेस बनाने का फायदा ये है कि वो फाइल कंप्रेस हो जाती है तो जल्दी रन हो पाता है क्योंकि सर्वर्स में जितने भी सीपीयूज हैं हम उनको एक साथ यूज कर सकते हैं जब आप एक फाइल को डेटाबेस में कन्वर्ट कर देते हो अगर आप कन्वर्ट नहीं करते हो और सिर्फ इंडिविजुअली चलाते हो तो सिर्फ वो सिंगल सीपीयू यूज करता है क्योंकि उसको पता है कि सिर्फ ये एक फाइल ही है उसको सिर्फ इसके साथ ही करना है तो हम उस पे थ्रेड्स नहीं बढ़ा सकते तो जब हम एक फाइल का डेटाबेस जल्दी सर्च हो जाता है हम किसी भी फाइल का डेटाबेस बना दे तो हमारा जो रिजल्ट है वो बहुत फटाफट आ जाता है तो आप अभी देखोगे कि हम एक डेटाबेस बना रहे हैं उसके बाद हम ब्लास्ट एंड रन करेंगे
डेटाबेस ये जो फाइल्स अलग-अलग उनका मर्ज करके
आप सिंगल फाइल का भी बना सकते हो जिसके अगेंस्ट आपको रन करना है आप उसका डेटाबेस बना सकते हो जिस फाइल के अगेंस्ट हो रहा है बना
साइज बढ़ा रही हूं दिखने का कहां तक साइज का
चेंज कौन सा कमांड है
मेक लास्ट
ऊपर करो सी ये वाला इन्होंने कमांड लिखा है ऐसा सबने लिखा रन किया हो गया फाइल आगे हो गया
okay if this command is there or not you can check it via
आप जो भी फाइल का इनपुट आउटपुट जो जनरेट होता है वो साइमल्टेनियसली आपको यहाँ पे भी देखने को मिलेगा
these files have been generated so means we had generated a protein data like protein database first thing if this file contains something it will be in kbs or something like that if a like empty file of it or if your command didn't work fine this file will be there the name will be there but the file will be empty so 0 kb will be there so please have a look on the kbs as well okay so next thing that I will be doing is
सी अभी हम टर्मिनल पे ही ज्यादा फोकस रखते हैं आप जितना विनएसएपी यूज करोगे आप लिनक्स में एकदम यूज्ड हो जाओगे जितना आप इस स्क्रीन को फेवरेबल रखोगे कि हां मुझे इसी पे काम करना है आप जल्दी यूज्ड हो जाओगे तो आपको बार-बार देखने नहीं जाना पड़ेगा कि हम कहां रन कर रहे हैं क्या फाइल का आउटपुट आया सब इसमें पता चल जाएगा तो हम इसके आगे अभी यूज्ड हो गए
okay so whatever file we obtain from any sequencing machine basically will be like what file we exactly want is FASTA or FASTA file okay or else other files like BAM files sample files are also there BCL files but we don't require them and we don't have them in our processes in normal processes in some other work BAM file may be in use so for this FASTA file will be generated as we are working on torrent data a single file will be there in Illumina data, 2 files will be there of same sequence for forward and reverse. Okay. So in iTorrent, a single file will be there, so we don't need to merge files. Basically, what happens is when we work on Illumina, the 2 reads will be there. So 2 files will be there. we need to merge them together first, and later we can proceed them for the analysis. In torrent, we don't have such kind of thing, so single file will be there. Moreover, FastQ file we had converted it into FASTA file. Okay, so FastQ to FASTA converter is there. We can convert our file, and next what we will be doing is we will be blasting our file against that database which we had created. Okay, so command is, as we are doing nucleotide blast, so blast N. Toolkit will be there available, so you need to download toolkits, but it has already been in our server, so we are not downloading any kind of toolkits over here. So blast N is for nucleotide blast. Blast X will be for nucleotide is your query, and the subject will be a protein. Then you will be doing blast X. Okay, so we are searching the whatever file we obtain is in nucleotide, and we will search it against nucleotide database. So we are having blast N over here. Blast N space. Now you need to have, see, if you want to know what exactly this file, this blast N will do, type -H, and it will show you a range of list. Whatever commands you can give, it will be there. See, blast H, blast N, and it has so many commands like H for help, import, search. All these are commands which we can type after blast N. If you need detail of this command, type -help after your whatever command. In Linux, it's best thing that whatever command, if you're stuck in command, you just need to know the name and type -H. It will give you the command which you can type after it. If you need the manual, type man. It will give you the manual as well. So for it, as we can see, what we will be working is, as we had created a database already, so we don't need subject file. As I just now told you, we can directly blast our files against FASTA file if we don't want to make databases. So for that, our reference file will be our subject file. But now we have database, so we will be having database thing. So for this, these all many things are there, and for all of your convenience, I had already I have a soft copy of this, and you can read it what these commands exactly are. So for now, blast N -query. It will be your sequence which you want to analyze. Okay. So just for your convenience, I'm showing it over here that we have S1 FASTA, S2.FASTA, S3.FASTA, and so many till S12. You can choose any of your FASTA file. Like just type over here, I'm going to choose S10.FASTA. So I'm going to work with it. Choose any file out of these 12 files.
Use tab.
Please make sure that you use a FASTA file, not FASTQ files.
आप फाइल का फर्स्ट 2 वर्ड लिख के टैब करोगे तो ऑटोमेटिकली वो आ जाएगा.
Has everyone reached? Everyone had typed their query sequences? Okay. So next thing that we will be adding is database. So DB. DB is the command for database. -DB.
तो यहाँ पे आप अपने सब्जेक्ट को भी स्पेसिफाई कर सकते हो. इसके अगेंस्ट रन करना है. जो मैंने बोला कि सिर्फ एक डिसएडवांटेज है कि वो 1 सीपीयू यूज करेगा. तो हमने जिसके अगेंस्ट रन करना है उसका हमने डेटाबेस बना दिया. तो अब हमारे सारे सीपीयूज यूज हो सकते हैं. तो अब हम डीबी लिख के हमारा डेटाबेस का जो भी फाइल का नाम है वो हम यहाँ पे रख देंगे.
And now just type capital N and tab. It will automatically take your database file. Capital N and tab.
Tab.
Full. Has everyone entered their database? Okay. Next thing in our command will be, see, as we run a local blast against any sequence of 16s or whatever, we obtain hundreds of numbers of alignment for a single file. what exactly NGS does is we have thousands of, even more than thousands of files in it. If we obtain hundreds of alignment for every file, we will be having a very huge file and ending up with a huge data, which will be of not of interest. So we have a command over here, number of alignments. So that will set how many alignments do you want. You can have one, you can have two, or you can have five, or so more. So for AMR analysis, we generally prefer one alignment because we just go with the topmost hit. So NUM is for number underscore alignments. And now I'm typing one. I just need one alignment per query. If you want more alignments, you can add numbers over here, but that will end up with a huge data which will be difficult for you to analyze. Done with it? Next command that we will be having is for our E value, expect value. What exactly we do is we want a higher E value. Means in minus range it should be higher, or in technically that I should be saying E value should be as low as possible. For our analysis, we use 10 raised to power minus 5 E value. So why we are like having this kind of command, so that the E value which are in positive or zero or something like that, that will not be in our results. We want to concise our results to somewhat that we can analyze, and that will be of our interest. So I'm typing now, command is hyphen E value 10 E minus 5. Because this is what I want as my E value. Done? Okay, so next thing that I want is percent identity. I hope like we know about percent identity. What exactly happens is when you have a query sequence and a subject, like whatever, like I try to match my hand with another of my hand. So what will happen? Some of the, like, this hand might match with this or may not match with this, but there should be a limited percentage. Like I want 95% identity, I want 30% identity, I want 50% identity, or what kind of identity do I want? Basically, percent identity in our case is set to be 95%. Why we are having this much stringent parameter is because we are working with a panel. The library has been created through a panel, AMR panel. So specific genes are to be amplified, which are of AMR only, related to AMR only. So we are keeping our like parameters very stringent in order to get only first hit and very limited data which we want. And even after this, you will see how much amount of data we will be getting. So next is percent identity. Percent identity. And now I am having 95%. Is everyone done till now? See, you can always add many more options in it, many more commands in it, but I'm just telling which are mandatory. Like you cannot skip any of these commands. Okay, next command which we are going to enter is for our out format. What kind of output do we need? See, basically, blast, okay, we will be having a blast result, but we are going to analyze it even in Excel as well. Because you will see, like in our blast, we have, for NGS, we don't have a single fragment for each sample. Like for our query, same query will be there multiple times. So we will be having same hit. Like for example, I have this remote as my query. It's my query sequence. Okay? I will not be having single remote. I am having multiple remotes at my time. Like the number of G will be there, so it will be in multiple copies. it's not necessary that you will obtain a single read out of it. So what will happen is the result which we will obtaining after it, it will show that okay, this is ampicillin resistant, this read is ampicillin resistant. But you are having multiple copies of this particular read. So you will be having multiple AMR like ampicillin resistance in your Excel data. So in order to analyze it in downstream, we first need to have this data in our Excel format, as in tabular format. So out FMT6 is there for it, and basically other formats are also there, which we have a README file in it, and I will share the file with you. So it will show you other type of out format which you can use. But for AMR analysis, we use out format 6, not for AMR, basically for most of the analysis, because it will give you a data in tabular form. So just write out FMT6. Out FMT is out format 6. What will happen after it? The whatever command I have typed over here, the result will appear on screen itself. If I press enter just now, the results start appearing over here. And please don't press enter. Okay. Whosoever had press enter, press control. Okay. So press control Z who had entered. And for that, there is a up arrow. Press up arrow. It will show you the previous command which you have entered. Okay. So you just now see what will happen if we just enter it. So what we will be doing is the output which will be generating from this file, we will be having that output in a text file so that we can analyze it later. So for that, add a greater than sign and name of file. Like S, for my file, I'm just giving s10.txt. File should be in txt format for now. Please give whatever, give your name. Replace हो जाएगा. Write your name in the.
आप अपना नाम लिख दो और .txt कर दो. नाम लिख के txt कर दो. Twinkle.txt.
Write your name and .txt.
अपना खुद का नाम. Greater sign is for whatever the input that you are giving. It will be generated in this file, twinkle.txt.
We don't want our output on the screen. We want our output in that particular file.
तो हम वो greater than sign दे दें.
हाँ, hyphen out. You can do that. So done with the command. You can add or you can remove. Press enter. If this blinking dot is on the left hand side, means your command is working. It will work. Everyone should have their like that dot on the left hand side. If it is on the right hand side, means there is a problem.
Yes.
Okay. Okay, one thing that we have done is we haven't given any kind of threads. So one CPU will be used in it, and as for from this speed, it will take a day to complete. Okay, so just press Control C again. It will kill your command. Press upside arrow. And add over here num underscore threads and add 10. Num underscore threads. Number of threads. Num underscore threads. Please provide 10 threads only each. No, what? Totally. Actually, it will start running, but it will not end. So if you want result tomorrow, then it's 128 in this server. Please provide 10 threads only. And even with this, you can assume that we are 11 people working over here, so 110 CPUs are working. Have my second. If you have that block on the left hand side, your command has been undergoing, so it will be finished within 2 or 3 minutes. नहीं आज ही बताओ ये आज ही होगा उनको 140 threads चाहिए अभी Okay, so meanwhile, this completes. Let's have a look on the web-based tools.
अभी ये रन हो रहा है, ठीक है। अभी जिस सर्वर पे आप हो, हम एक्चुअली हमारे पास जो सुपर माइक्रो है, वो it contains 228 threads, 228 threads। तो हम सारा का सारा नहीं यूज करते अभी, क्योंकि हमारे सुपर माइक्रो में बहुत सारे वर्चुअल मशीन्स बना के रखे हैं, विंडोज और लिनक्स, जिसमें हमने 30-40 थ्रेड्स बना के अलग-अलग वीएम बना के सबको यहाँ पे अलॉट किए। कि जितने सारे प्रोजेक्ट्स हैं उनके अलग-अलग हमने उनको वर्चुअल मशीन्स बना के दिए हैं। तो मैक्सिमम नंबर ऑफ थ्रेड्स जो हम बेसिकली देते हैं उनको, वो 30 ही देते हैं। तो अभी जो आप ये दे रहे हो, ये इट कंटेन्स मैक्सिमम नंबर ऑफ थ्रेड्स ऑफ 30। तो हम दूसरा एक वीएम दे सकते हैं अभी, जिसमें हमारे पास बहुत सारे थ्रेड्स हैं, 228, बट अभी के लिए हम इसको यूज कर रहे हैं। देखो सी, अगर आपको कभी ऐसा और हम लिनक्स पे डीप में जाएंगे, तो कभी देखना हो कि आपके पास सी इसको बड़ा कर।
अच्छा 32 ही है इसकी।
तो ये जो अभी आप यहाँ जो सब आपने रन किए हैं एक साथ। तो ये जितने भी आपको दिख रहे हैं ब्लास्ट एंड क्वेरी, ये सब अभी रनिंग में है हमारे पास। इस वर्चुअल वीएम में जो आपने जिसमें लॉगिन किया है उसमें सिर्फ 32 थ्रेड्स हैं और आप सबने 10-10 दे दिए हैं, तो इतने तो है भी नहीं इस वीएम। तो बट चलेगा, ये आउटपुट आ जाएगा, आपका थोड़ा टाइम लगेगा बट आ जाएगा। तो ये आप देख सकते हो करंट टाइम कि आपका कौन सा कमांड रन हो रहा है बैकग्राउंड में। अगर आपको किल करना है तो आप यहाँ से भी किल कर सकते हो उनको। ये जो आपने कमांड रन किए वो आप यहाँ पे देख सकते हो कि ये अभी कमांड सारे रनिंग में है सबके सब।
Okay, so meanwhile, we can learn about the vacuum. Basically, until this command finishes, we can learn about that. So the first web tool which we will be saying is OneCodex. So it's available. You can create an account over here, but as I already have a OneCodex, one CODEX. You can easily create a free account in it, but I already have an account, so I will log into it. So this is how it will look like. Over here, a name is there, upload and import. You can directly drag your file here. Your FastView file will be the input file here. So you can drag into it, or FastView and FastDraw, both can be included. So just for now, I'm showing you it for a file. By this, you will also come to know about its limitation. That's why I'm doing it. The file will be uploaded. Like I just clicked on the browse my file from the Windows. The main limitation that we are going to encounter in it is it will only show us the diversity of microbes. Like whatever microbes are represented in this particular sample. We are not going to have AMR analysis for free. It will charge approximately $20 per analysis. So, like up to what you can do is I'm showing it to you over here. Or else the only web tool which works for free is Rasfinder. Why I'm showing you this is if you are able to pay for your data, then OneCodex will give you a whole range of lists. I have some data of mine which has been analyzed through OneCodex. I will let you know. The Pruna charts will be there in its result itself. So that will be good to see. Like you will not be having any visualization tool after it. Because whatever analysis we are going to do in it will end up with a tabular form. Like this many genes, resistant gene, and this many number. The downstream analysis, the representation for your papers and for your work is to be done, which will be shown by Dinesh sir in STAMP. You can do analysis in STAMP as well. But in OneCodex, they will do the analysis, they will even give you a graphical representation of it as well. So over here, the data has been successfully uploaded. In samples, you can see this file. They itself will analyze it. What exactly it is a metagenomic sample. So no community is in this abundance form, or none other community is in, like, if I am having a particular strain of bacteria in which I am analyzing my AMR, that particular DNA or that particular bacteria should be most abundant bacteria in my sample. That's the case which Martin ma'am even told, that we should have a pure DNA sample. If not, like for this metagenomic sample, we don't have a particular DNA, like we have a range of DNA. So in it, no community will be in its abundance. As you can see over here, it's a sample from Klebsiella pneumoniae. So it's having a tab, like a tag is there, Klebsiella pneumoniae. So the sample belongs to the bacteria Klebsiella pneumoniae. It's the most abundant bacteria. It's some other analysis which I had done. For metagenomic sample, you will not be having any kind of tag. This iron over here show it's like what kind of platform have you used. Like over here, you can see Illumina MySic because it's a Illumina data. So it will be completing. See, you can have complex when your sample isn't pure or you are analyzing a metagenomic sample. The tag of complex or maybe no other tag will be there. But if it is a pure sample, isolate tag will be there. The tag will be given by the system itself. It's in pending. It will take some time. Over here we have run analysis. In run analysis, it's directly asking for payment. See, one codex database for metagenomic classification is what we are doing currently because it's free of cost. This AMR panel and Escherichia coli panel cost $20 per sample. So for that, like it's very difficult for anyone to afford it. For per sample, you need to give $20. Let me just show you what kind of, because the same file has been run by me earlier, so I will show you what results do you obtain. This high abundance means it doesn't contain the, like, no enrichment was done, no particular bacteria was tested in it, so no bacteria is there in its high abundance. Medium abundance and low abundance, like basically it's showing class or bacteria whatever are there. But we cannot see, to be honest, we cannot rely on this. Why exactly? Because for AMR, we have small strand of resistant gene which are to be amplified. On the basis of those small strands, it's showing us the abundance. So those strands, like small strands, aren't even 16s. They are just random strands, like not random. Basically, they are antimicrobial resistant genes. So according to that, it's showing the abundance. But it's not the correct metagenomic analysis which we can do. You can add your 16s sample over here. So that will for sure give you the correct abundance analysis. It's giving you a table as well. So over here, you can choose what kind of level. Like if I choose family level, it will show what family are there, what families are there. Order, class, phylum, species level. Okay, so up to this you can do. And in node, you can see like 5410. It will show the nodes up to like 5410 should be the minimum number of reads in that sample, in the R sample. Those nodes are there. If it so you can reduce the number of reads from here, and a lot of other nodes will appear like this. So through this you can do one codex analysis, but for AMR you have to pay. Okay, it's over here, whatever you need to pay. I will just show you the results which we obtain after the one codex analysis, paid analysis basically. First type of Prona will be output apart from the table. I will show you the table as well. So it will analyze all the type of like databases which you want and will give you an output in such form. Like from Rasfinder, it has shown that step to like this macrolides are in highest amount with 7% ERMBG and like 4% LNC, tetracycline W. So such like through AMR finder, basically every database will give you different results. You cannot compare two databases. Like if you want to compare, the results will be different. There is no surety you will obtain the same results from both. Even you can see it from here. The NCBI is the largest database, so it is showing so many results from the same sample. But one codex is not like the codex itself have its database, so it's just giving these many hits only. For AMR Finder, we have these many, and for RAS Finder, we have these many hits. So it's what Trona is about. Like one codex paid will give you like results like this. Next software is Rasfinder. So you can do it for your sample and it is free of cost. What exactly it shows? Chromosomal point mutation. If you click on this, then it will show the AMR which are on the chromosome. Like only chromosomal gene will be counted. If not, if you click on acquire antimicrobial resistant gene, then it will give you even a class of gene which you want to check. If you want to select anyone, then you can do it, or else you can just don't select anyone. It will by default take all the classes of antibiotics which are there. So select threshold for percent identity. What exactly it shows? Like how much percentage identity do you want to check from your sample and the database? And minimum length that is to be matched is 60%. Like why we have such kind of minimum length concept is if I have 5 base pair sequence, for example, and out of them only 4 base pair matches somewhere, then it will show 80% identity. But that 80% identity we don't want it over here because we want that minimum length should be 60%. 60% match should be there. You can increase it if you want. So select any species. You can do it against a particular bacteria only. So it's the limitation of this thing. So I am running it against Klebsiella. Next is what kind of read do you have? If you had assembled your read, then you can do assembled genome contigs. If not, 454 Rush system was there, which was used for NGS earlier. Later, like currently we use Illumina and ion touring data. And even in Illumina, if you have single end chemistry, if you had used or paired end chemistry, you can select it, solid and ion touring data. You can choose any of these. So I already have assembled genome. So I will choose it and choose my file from the database. And click on upload. It will upload your file. As soon as the file uploads, a new tab will open. If you want to work with multiple files and if you you have a lot of work, you can just directly type your mail ID over here. It will send you the link of the result. So you can type it. And if not, then the same page will reload and result will appear on the same page. So we will just wait for the results. Let me just check if our blast has been completed or not. देखा आपने? Okay. Our blast has been completed. Anyways, let's wait for this result first. And okay, let it be. I just add my mail ID over here, so it will send notification. Meanwhile, all of you, please check whether your Kuti has completed the command or not. The blast has. If the cursor is on right, means the command has been run successfully. So in WinRecipe, if you refresh it, you will be having your name. The text file which you had created will be there. Please have a look on your output file. Is it there? And it should have something in it. It shouldn't be of 0 KB. Okay, so on left hand side, click on the desktop. So you will directly go to desktop. Click over desktop here on this side. Now we need to have downstream analysis, so click on desktop. This desktop should be on desktop.
अपने लेफ्ट वाले विंडो में ड्रैग कर लो कंप्यूटर इंटरफेस है वो सर्वर पे है
Be on desktop.
बट जहाँ ड्रैग कर रहे हो वहाँ पे एक के पास
You are on next one. Is everyone able to be on their desktop? Now just drag the file which you want to analyze onto the left hand side. Just drag and drop.
Yes, अपने drag and drop कर
Drag and drop. Okay. Now you can just minimize it and see whether your file is on desktop or not.
आप अपने पीसी में आ जाओ और देखो वो डेस्कटॉप पे आई है वो फाइल या नहीं.
See, I have this file over here, twinkle.txt. Everyone should have their file on the desktop. Now, right click on it and open with Excel. Open this file with Excel. Open your file with Excel. The topmost column, like insert a row over here. Insert a row for the headers. As it doesn't contain any header, we need to have a header. So insert a row. So what exactly it means is you can literally Google it. It will show you. Because every time you will be doing it, it will not show the headers. You can type blast out FMT6 results and it will show such kind of screen in which you can have your outcome and what headers they have. Blast out FMT6 headers. So these are your headers, default values are. So you have to copy it, so please search it. Blast out FMT6 headers or results. It will show what default headers are there. You can add headers manually even. Just copy from query sequence ID to bit scope. Copy it and paste it over here on your text, like Excel file. Done. Now you can just click on data. There is a function text to column. You can use it and it will separate your data in the row column itself. So just use the function text to data and finish it. So your first header. Now data text to column. Next, it will separate it like this.
Can also define in as a message running, but अभी सिर्फ आप हेडर आपको सबको हेडर
I hope you are done. Has everyone reached up to it? Okay. So now the first column is our query sequence ID, Q set ID. Okay. It is like automatically generated some kind of ID which is given to our like if we had opened our FASTA file, it should start like any FASTA file should start with a name greater than and a name will be there, and in the like in below section there will be the like nucleotide sequence. So this name basically are our query sequence, which exact like every FASTA has a name in our FASTA file. So these are our query sequence. Next is a subject sequence ID. So please have a look over here carefully. We have set 4, which is our gene, antibiotic resistant gene, and Campylobacter. See, basically what STAR database does is, in this database, it shows the probability like, okay, this gene has been arose from Campylobacter. Like SAT4 is from Campylobacter. It has already been there in its database. We had done nothing in it. So for result, it has shown that this query ID, this particular XG79, something like this ID, belongs to set 4, that is AMR resistant gene, which is of Campylobacter. Okay. So next is percent identity. What percent identity does it have? So as in our normal class, we have percent identity. It is similar to that. It shows what percent identity it is, the length of query that has been matched with the subject sequence, mismatch, how many mismatches are there, gap open, if there is any gap or not, query start, query end, from which nucleotide the query has been started and to up to what nucleotide it has been ended. Subject start, subject end. Must mean the subject length should, like was higher and your query is of shorter fragment. So on what nucleotide of your subject the query has been matched. So it's subject start and subject end, e value and bit score. Based on any information you can obtain. Even if you want to have more of information, you can always add out of empty and whatever information you want, you can then choose it. So it will give you the information. So it is not something like we can have an like it's not some usable data because we cannot do anything out of it. So now what we need to do is pivoting. It's in Excel basically. What exactly it will do is first we will separate the AMR gene in a column. Second, we will have the corresponding bacteria. So one query will be there, a query ID will be there, a resistant gene will be there, and an organism will be there. These many data will be useful to us. No doubt, it shows an like if we even need to reduce our data, we can always increase percent identity from here by removing others. But for now, as I already had said, 95% identity, that is okay for me. So just copy column A and B and add a new sheet over here and paste these two. You just don't need to select whole, just select the headers of the column. Column 1 and column A and column B, it will select the whole row itself, like whole column itself. And Control C, add into the new sheet. Is everyone able to reach up to it? फर्स्ट टू कॉलम को नेक्स्ट शीट में पेस्ट करना है. Not to drag all the column, or else it is something 80,000 entry. please don't select column manually
You can select from the top.
because these may be 80,000 or more entries. So it is not feasible for you to drag all the way. Just select from here, A and B. It will just do the work. Okay. Now click on the column B only, as we don't need the other data. We just need the antibiotic resistant gene name and bacteria name. So data, click on data over here. Select the column B first, the whole column B from here. Now click on data and text to column. Text to column. Now click on next. Over here, delimiters are there. So click on others, untick tab. Click on others and add pipe sign over here. Why we are having pipe sign is our data is separated through a pipe. That's why we are just separating our data in different columns, nothing else. So next, it will show you what kind of data you will be obtaining and finish it. So by the end, you should be obtaining such kind of thing. Delete rest other column except column G, maybe that contain your antibiotic gene and bacteria. So delete rest other columns. You don't need them. Delete rest other column except gene and bacteria.
ये वाला स्टेप वापस बताना क्या डिलीट करना है
बाकी सब कॉलम डिलीट कर देने हैं बस वो बैक अच्छा उल्टे कंट्रोल Z हो गया ये सभी बैक अरे
लास्ट कॉलम
हाँ लास्ट टू कॉलम
वहां से फॉरवर्ड कर दो क्या रखना है और क्या डिलीट करना है
बाकी सब
सिर्फ लास्ट के दो कॉलम रखते हैं
please keep last two columns only. This set4, tet2, tetw, these are your resistant gene and column next to right shows your resistant bacteria. So delete rest other columns and keep these two only. Write gene and bacteria on it. Okay. So now it was general Excel. Now what we will be doing is pivoting. What exactly pivoting will be doing? Will do. See, you can see tetracycline W over here, tetracycline W over here and here as well. There are lot of entries of tetracycline W, even other gene as well. So what we will do is we will have like numbers. Basically tetracycline W, these many queries belongs to tetracycline W. We will just have number because this query ID means nothing to us because we are not gonna see each query ID. We just need how many numbers of tetracycline resistant genes are there. So for that we use pivoting. For pivoting, just select first two column. For that, control shift. Use control shift and down. Why I'm saying it? Because as in my column it has 8,62,000 reads. Like 8,62,000 entry. I'm not gonna drag and drop. Control shift and down. First two columns are to be selected only. Don't select the column of bacteria. Not a phone. See, even sometimes it please press Control Shift down, alright, or else it have 8,62,000 entry. It will be too long to drag it down. See, even with this much stringent parameter, we obtain such high entry for a sample. If you keep the parameter low, then entry will be more. So it will be more difficult for you to analyze. I hope everyone has reached up to it. Now, please go in insert. Just a minute. In insert, we have a function of pivot table. Please click on pivot table. It will ask you to delimit your area, but we had already done it by selecting insert and pivot table. Now click on OK. A new sheet will open. Okay. Has everyone reached up to it? Akash sir. Okay. Okay. Now these gene name, we want them as in name. So drag this gene and drop in row. Drag the gene and drop in row. So as soon as I drop it, the name of gene will appear on the left hand side. Drag it, just drag it above, drag it from upside down. Now query sequence ID. Drag this query sequence ID and add in values. It will give you value. So these resistant genes are in this much number. That's what this table will tell you. And dropping down. Is everyone able to do it? I'm deleting the above two row and just filtering it. Okay. You cannot change anything in it. So copy this table, like Control Shift, copy it and paste in the same sheet as values. Over here as values. You need to paste, like copy this whole table and paste as values, or else you will not be able to manipulate the things. Copy this table and paste as value in the same sheet. Chalan? Paste as values. Over here we have value option. Paste as values. The blue color, like the blue shade at the top, should not be here in this. Now you can add a filter on it. By filter, you can just largest to smallest. You can sort it from largest to smallest. And you filter it from the count. Count table is there. To filter it from here. Okay, so what exactly it is showing? See, exactly it is showing that total there are 8,62,378 reads. Like these many queries were there. Tetracycline W was it the most abundant resistant gene found. It was about 2,46,198. Why we exactly need it is like you don't have to go with individual number. We need to normalize our data first. And why we need normalization is see in one sample I have 5,000 reads, and in another sample I have 1 lakh reads. Okay, so in 5,000 rate, if I obtain 400 tetracycline W, that will be even considerable because the total number of rate was 5,000 only. But I have 400 tetracycline W in 1 lakh rate, that will be negligible. However, the resistant gene was same in both the samples. So it is not okay to like have a percentage. It is not okay to go with the exact count. We should always normalize our data. It's like which has been told in our bachelor's and master's as well. So for normalization, you can do two things: either take log or take percentage. So you can any way is fine. You can either take percentage. So for percentage, you have to divide whole the data with your take the percentage from the grand total. That's why I had kept that grand total it above. So you can directly write in formula in Excel to see I show you. You can use this formula. What exactly it is showing is this formula E2 cell multiply by basically it's percentage formula only. It will calculate the percentage.
We are converting this number into percentage.
After that, how the representation will be done, that will be done on SAM. But you need to normalize your data in Excel before taking it for further analysis. Never ever do the analysis with your raw data. It will eventually and you will eventually end up with the wrong estimations. And you can just double click at the right hand side and it will show the percentage of all the others. From here as well, you can directly whatever number you want to take, this number multiply by 100. So this is the way we calculate percentage and divide by the total. It is showing 100%. If you click right, you can go up here and double click it. So for that, now you need to have pivot. So I have selected all the samples. Insert pivot table. So please make sure over here number one is A block is already in this column should be there. In most of the case, this column will be there. So A block will be there. Okay. Now gene should be your row. Query should be your value. From now you just need to copy it and paste it like here onwards you need to filter it. Filter it and make percentage of each sample. See, for most of the analysis, what data but what basically we consider is more than 1% rate should be there. Like more than 1% resistant gene should be there of that particular gene. So over here, APS2 28 was in 1% and rest other was in 0.6 percentage and something like that. So we cannot consider the things below that. If you want to consider, you can consider, but that will make the graph basically messier. So 1% is okay. Even if you want to reduce your, like, increase your percentage, you can even do it by this. Because after that, it's just for a sample. You need to do it for all of your samples, and later you can plot it in graph. A single sample cannot be plotted. So for every sample, you have to do something like this, and I have your 1%, maybe 1%, maybe more than that. This is the result which we had obtained for Rasfinder. See, it's showing the antimicrobial, its class, and prediction, whether it is resistant or not. You can obtain this from Rasfinder directly. So as it is showing resistant, it will even give the name. So at the downside, you can have a table. A complete table will be there. And this table will show that how many resistant genes are there, and it is basically like divided from the class. It is a Rasfinder output that we are going to obtain. So in Rasfinder, you will be obtaining outputs like this. And as you can see, it's just giving few outputs. But in Ras, we are getting a lot of output because of change in databases. You cannot compare to databases. So this is how Rasfinder works. And this is how we had completed our analysis for like last Linux-based analysis has been completed till now. Now, how to visualize this data will be on STAN. You cannot like you can create graphs from here, but that will not be efficient, and you can only get bar graph or something like that. But in STAN, you can have more of statistical analysis. So that will be taught by Dinesh sir in next session. So if you have any query till now, please do let me know. Now we will be seeing, like in next session, we will see how this table and how this table can be viewed using STAMP and statistical analysis over it. So any question? I hope you guys understand. Okay. You guys can go for the ID, and later we will meet again over here to analyze this data and stat. For next session will be statistical analysis. Like we have 12 samples in that. One sample might get like that.
रिकॉर्डिंग स्टॉप कर देना.
Like your sample contains 50% of heterosexual W, means 10,000 genes of heterosexual W was there. And 20,000 means like 20,000
करना है, स्टैम्प करना है.
Recording stop.